What is DNA? DNA is a self-replicating material which is present in living organisms and is the carrier of genetic information. This means that the DNA in your body is what makes you you and different from everybody else. DNA is made up of 3 different parts. When these things combine to make a strand of DNA, 2 strands combine to make a double helix. This is like if someone took a ladder and then twisted it. If the 2 strands of DNA make the ladder, then the nitrogenous bases make the steps of the ladder. The base on one strand of DNA bonds with the base on the other strand of DNA. Now that we know what DNA looks like, we're going to look at how it duplicates. When DNA replicates, the first step is for the DNA to unravel. This is like a zipper of DNA unzipping. Once the DNA is unraveled, it splits up into a fork. This is where something called an enzyme can help with the DNA replication process. The enzyme moves across the strand on the top and makes a continuous line of DNA. On the bottom, DNA is made in little sections and in the opposite direction. Now there are 2 strands of DNA that work in your body. Now that we know the basics, we can look at DNA and the replication in more detail. Remember the complementary base pairing we talked about early in the video? We can now look at the 4 bases, which are adenine, cytosine, guanine, and thymine. Adenine bonds with thymine, and cytosine bonds with guanine. In the DNA molecule, these nitrogenous bases are held together by hydrogen bonds. When DNA is ready to replicate, the enzyme DNA helicase goes over the strands of DNA and breaks the hydrogen bonds between the complementary base pairs. This leads to the formation of replication forks, which combine to create replication bubbles. Now we can take a closer look at the replication fork and look at how DNA is replicated. On the top, there is something called the leading strand, and on the bottom, there is the lagging strand. On the top, an enzyme called DNA polymerase III moves over the strand and adds the missing bases to the base pairs, A with T and C with G. The lagging strand works in a similar way to the leading strand, but it works in the opposite direction and in smaller fragments. There are now 2 more strands of DNA which automatically twist into a double helix. But that's not all there is to it. We know that DNA, or deoxyribonucleic acid, is made up of 3 parts: a deoxyribose sugar, a phosphate group, and a nitrogenous base. We know that there are 4 nitrogenous bases: adenine, cytosine, thymine, and guanine, where adenine and thymine and cytosine and guanine are held together by hydrogen bonds. In DNA, the phosphate group, the deoxyribose sugar, and the nitrogenous base, which can be a purine or a pyrimidine, where the deoxyribose sugar and nitrogenous base are held together by a glycosyl bond. Together, these form the strand of DNA which runs anti-parallel. This means that one direction runs from 5' to 3' and the other 3' to 5'. DNA replication also occurs semi-conservatively. This means that each strand of new DNA molecule is composed of one parent strand and one newly synthesized strand. This makes the process of DNA replication semi-conservative. DNA replication starts when an enzyme called DNA helicase comes in and breaks the hydrogen bonds and unravels the double helix. Now that there are no more hydrogen bonds, single-stranded binding proteins can attach to the single strands of DNA. These proteins are what prevent the hydrogen bonds from reforming and allow the replication fork to stay open. Next, an enzyme called DNA gyrase comes in and relieves any tension that is still formed between the unraveled strands of DNA. This creates a replication fork. Over time, when two replication forks occur close to each other, a replication bubble will eventually form. Next, an origin of replication at the top of each bubble will form as well. Now, a temporary RNA primer attaches itself to the single strand of DNA. This is important because the enzyme DNA polymerase III cannot initiate a complementary strand on its own and relies on RNA primers so it can add deoxyribonucleic triphosphates in the 5' to 3' direction. On the lagging strand, something called Okazaki fragments occur, so DNA polymerase III can work in the 5' to 3' direction, for the lagging strand naturally runs in the 3' to 5' direction. Once this occurs, DNA polymerase I can now remove DNA primers and replaces the appropriate deoxyribonucleotides. The last step of DNA replication is for the enzyme DNA ligase to catalyze the DNA fragments together by forming a phosphodiester bond and sealing the strand of DNA. Once again, this leaves us with DNA fully replicated.